We're very excited to have Dr. Eric Doucette speaking this evening. He is currently an assistant professor in the biology department at the Massachusetts College of Liberal Arts in North Adams, Massachusetts. I've recently learned he was just approved for tenure this past month, and so he'll be an associate professor at MCLA this coming academic year. So we want to extend our congratulations to Eric. At MCLA, Eric teaches courses ranging from field botany to conservation biology and ecology, as well as bryology and lichenology. And many of these are new to the college and bring an invigorated botanical element to the biology curriculum. So we're very excited about what Eric is teaching and bringing to students at MCLA. Prior to starting at the Mass College of Liberal Arts in 2016, Eric earned his PhD in ecology and environmental sciences from the University of Maine in 2015, completing his dissertation on the systematics of agamic complexes in Amelanchier, working with Dr. Christopher Campbell. Eric also worked for many years as a botanist and ecologist for Stantec Consulting in Topsham, Maine, undertaking natural community and rare plant and animal surveys. So he has extensive field experience in Maine and New England. Eric has authored and co-authored a number of botanical publications, including research papers in the American Journal of Botany, Systematic Botany, and Rhodora. And he also co-authored with Christopher Campbell the Amelanchier contribution to the flora of North America in volume 9. Dr. Doucette has been active in the botanical community in New England for many years. He's currently a member of the New England Botanical Society's Council, and he also served as president of the Joslin Botanical Society in Maine from 2010 through 2018. This evening, Dr. Doucette will be presenting a talk entitled "A Species Complex Approach to Taxon Delimitation in Amelanchier Shadbushes." So we're excited to hear from Eric and hear more and learn more about Amelanchier in New England, and the timing of this is perfect. With many of these species in bloom in recent weeks. So without further ado, I will turn it over to Eric to tell us about Amelanchier.
All right, thank you, Jesse. Scary sounding title of a talk, isn't it? So as Jesse mentioned, I do teach at a college in Western Mass, largely plant-related classes. I'm very lucky in the fact that I'm the only field biologist in the department. It's a very human-centered department with nursing programs, etc. So the upshot of that is I get to teach a lot of the plant classes that I want, and so it's been great. Prior to that, I have been really lucky to travel around a lot of the country and do some community surveying and rare species surveying, mostly in New England and the Maritimes, but occasionally further afield. As this picture in the lower right shows you, done a lot of work in California, mostly with Amelanchier, but there's a lot, as you know, the flora of California is very unique, lots of endemics. And so I've been lucky enough to spend some time there botanizing. And I still do a lot of work on the side, sort of pro bono work, I guess, if you will, for land trusts, doing natural community surveys and rare species surveys on their properties. I'm a big believer in sort of giving back to the public. Okay, so if you're interested in a plant group that's going to torture you mentally, then Amelanchier is the plant group for you. So like most botanists, I'm interested in plant groups that are taxonomically challenging. So some of my favorite genera, in addition to Amelanchier, are things like Solidago, Symphyotrichum, willows, the genus Salix. And those are all genera that have high degrees of hybridization, and it can be difficult to tell where one species ends and another begins. And so part of my, I guess, training in plant systematics, I became really interested in the philosophical underpinnings of what species are and what creates a species and how we define a species. And so it's worth noting that that philosophical underpinning is often at odds with field botany. And so it can be challenging, but also fun to try to reconcile those two different subfields. If you've ever looked at any paper in Amelanchier, I'm sure you've noticed that the very first line, the first paragraph, has some clause like what I have on the screen here, saying that Amelanchier is a horribly difficult genus, essentially, that there's problems with identification and problems with taxon delimitation. In other words, problems with defining what a species is in that genus. And that's certainly true. And there's real biological reasons for that, as well as sort of some historical reasons that have to deal with humans' interpretation of species concepts. If you don't know, and it is blooming right now, as Jesse said, this is a timely talk because there's lots of Amelanchier in the landscape that's blooming right at this moment. In fact, it's a little bit earlier this year, and the species that I have on my screen here in flower, Amelanchier arborea, that is in my house here in Western Mass. It's on the tail end, some of it's gone by. It started blooming April 19th, which is the earliest that I can recall it blooming in this part of New England. But it's a member of the apple subfamily of the rose family. And so that whole subfamily is well known for taxonomic problems, not just Amelanchier, but it's also used horticulturally. Lots of Amelanchier planted around people's yards, banks, shopping malls, etc., because it has this great floral display, great tasting fruit, makes a nice wine if you can get enough of the berries, great pie as well. And of course, it has a lot of wildlife value in those fruits. So just to give you some basic background, I don't want to assume that everyone knows this terminology. So there are different ploidy levels in the genus, and those ploidy level differences are at the root of some of the taxonomic problems. So the easiest way to think about this, or the easiest starting point, I should say, is that there are diploids in the genus Amelanchier. And that's like us, and in the sense that there's one set of chromosomes from each parent. And in Amelanchier, as in most plants that are diploids, they reproduce sexually. So they're obligate sexual species. And there's a high degree of self-incompatibility and very low fruit set in the absence of other reproductive individuals among these diploids. So in the same genus at the same time, though, we have tetraploids. So there are plants that have 4 sets of chromosomes, in this case, 2 from each parent rather than 1. And so the process of this genomic doubling is called polyploidization. And in Amelanchier, it almost is always to a tetraploid, a 4x condition. But just as an aside, in plant evolution in general, polyploidization has been hugely important. Entire clades of land plants have evolved through the process of polyploidization. In fact, the entire apple subfamily of the rose family did arise that way through a hybridization event that was coupled with the genome doubling event. And that produced the clade that has pome fruits, apple-like fruits, of which Amelanchier is one. So the cool thing about this in terms of plant evolution as a whole is that when you have these extra chromosomes, they're often free to do new things. And so the general genetic theory is that they're somewhat relaxed from strong selective pressure that the parental diploid genome might have been. And so that relaxation of selection and the allowing, if you will, of new traits, new morphologies, new ecologies, new pollination systems in some cases, that's called neofunctionalization. And that's been hugely important for plant evolution. And sometimes that's coupled with the new tetraploid finding a new niche, so partitioning the parental niche sometimes finally, in other cases occupying entirely different niches, niche shifting. And for plants, as Amelanchier among them, it's also been important for as a force for speciation in the group, and diversification in the group. And so in plants in general, plants at the tetraploid level, they can still be sexual, asexual, or both. In the genus Amelanchier, however, they're almost entirely asexual. There's just a very small residual amount of sexuality on the seed side, on the female side. But it's enough, 1 to 3% of residual sexuality, it's enough that back crossing of tetraploids to diploids and tetraploids among themselves is what can create these novel morphologies in the landscape, which I'll talk about. And then we can also have triploids. And these are, as we'll see, sort of a double-edged sword.
they can spur diversity, and they can also be a block towards further diversity with something called triploid block, which is well known in plant genetic systems. So for us in the genus Amelanchier, they're definitely a minority numerically on the landscape. They tend to occur where diploid species overlap each other and or where diploids overlap tetraploids. They're generally not locally abundant on the landscape, aside from a few geographical areas. Southern Oregon, Northern California tends to have a very high clustering of triploids. But by and large, across the North American landscape, they're fairly rare. And so they're oddballs, of course. They have 3 sets of chromosomes, 1 from one parent and 2 from the other. And they can be generated in a few different mechanisms, different ways. And so one of the papers that our research group, primary author of Michael Burgess in 2014 put out was an analysis of ploidy levels in the genus in North America. As you'll see going on, the genus does occur in Eurasia, but our sampling there is much smaller. So a lot of our data sets that we analyze tend to be from North America. It's a more robust sampling there. But and this is a lot of chopping. So if you're familiar with flow cytometry, it basically involves you sitting underneath a fume hood, having fresh leaves that you pour some chemicals on, propydium iodide, and you are chopping the leaves finely like a sushi chef. And you take the resultant chop bait, if that's a word, and you run it through a flow cytometer. And there are lasers that shoot through the suspended liquid that give you an estimate of ploidy level chromosome count using domesticated P as a standard. And so from the years of work, we have a really robust understanding of what percentage on the landscape nationwide, or continent-wide, I should say, are diploid, tetraploid, and triploid. And so some of you, I'm sure, know this, but my advisor, Dr. Christopher Campbell, who's on the lower left in this photo, he's been researching Amelanchier since a few years before I could legally drive. So quite a while. I'm 50. So he's been at it for a long time now. And it's been a very collaborative group working on Amelanchier over the years, including some other people that aren't shown here that have contributed as well. But from left to right in this picture, it's Chris Campbell from UMaine. In the back center there is Chris Fry, who's the state of Maryland botanist, who's state botanist for Maryland. In the center is Kevin Cushman, who's currently at Texas Tech. There's me in the back. And then to my left in the photo there, Michael Burgess, who's at SUNY Plattsburgh. And then a few years ago, Garth Holman, who went to UMaine with us, but is now in Michigan, joined the project. He's in that inset photo there. So the beauty of the collaboration, of course, is that we all are able to disperse every spring and collect like crazy, because despite it being a large geographical area, there's often flowering at the same time due to differences in elevation. So for instance, right now, we have Amelanchier arborea here flowering. It's also flowering at high elevation on the Southern Appalachians. So it was pretty hard to be in the Southern Appalachians and New England at the same time, obviously. And the same is true, but to a lesser degree, for Western North America. So we all kind of scattered around the country. And you can see here some of the stats of years of collecting data. This is a little dated now as well. So I haven't added on here the last few years of when I've collected plants, I guess a little more incidentally, but we're probably over 140,000 car miles. That does not include flying around the world. And Chris flew to a lot of old world sites. So we've definitely put some miles in. We've got a huge data set for morphological characters, a large data set for flow cytometry, and a large data set for molecular phylogenies. And all this data, of course, gets piped into our classification. And so the different ploidy levels in the genus that have posed problems have made us ask over the years, do diploids, triploids, and tetraploids, do they form different kinds of groups in the genus? In other words, are some species and some aren't? And the answer to those questions feeds into the systematics and how we're going to define the species in the group.
Just as an aside, in the lower right here, here's a photo that I took, that's a week and a half or so ago now, just down the road from my house here in Williamstown. And so the monograph, which I'm hoping, if it kills me, is submitted this year. Obviously, the ultimate goal is to have a classification of a global classification of the genus. But we have some additional goals which have been really important in framing how we approach the project. And so ultimately, sexual diploids are the ultimate drivers of diversity in the genus. So their genomes and combinations thereof and their hybrids, they're responsible ultimately for diversity. So we want to take and have taken a diploids first approach to delimiting species in this genus. Now, that's not to say that we're only recognizing diploids, but they have primacy, they're paramount. At the same time, we want to recognize widespread polyploid species that have a discrete morphology, discrete ecology, etc. And I don't want to say we're biased, but we are taking a little bit into account historical precedents. So there are some taxa, like Amelanchier levis in Eastern North America, for instance, which you'll see may sort of break some of our definitional rules, but it's been a long recognized taxa by all workers in the genus. And so that's been important to us to not totally upset the apple cart. And that's what number 4 is. We don't want to totally overturn the genus. We want to have a classification that's usable for the end user, but as best we can, has high fidelity to our data and the different types of data that we have. And so one of the things that we'll talk about that's problematic are so-called microspecies, and I'll kind of define those in a bit, but we want to recognize them in an informal way. We don't want to give them a formal name. And I think you'll see why. But we do want to draw attention to their importance in the landscape because, especially in New England, they're a large percentage of what you're going to come across. So we joke with each other over the years that, and there's some truth to it, that when we all started in Amelanchier, we all had a pretty good understanding of what species were in the genus. I mean, we thought we knew what all the species were. And in some ways, the more you study the genus, the less confident you become. And that's sort of one of the sad truths, I guess, with diving into any large complex genus. So in 2015, we published a paper, again, with Michael Burgess as the lead author, sort of categorizing these diploids in North America, where they occur geographically, their ecological importance, if they were differentiated ecologically, phenologically, cytologically, etc. And the goal with that was the diploids are a framework to then help us understand further complexity at other ploidy levels. And so this, again, is what's called the diploid first approach, which is becoming pretty standard, I should say, in plant taxonomy. So when we published that publication, the intent, of course, was to have further publications, papers and/or this monograph. And so we didn't formally name a couple of diploid taxa that we knew we were going to. And so that's why on the map, if you look at the Southern Appalachians, where you see A. levis and A. sanguinea, those are not the same that you might be familiar with here in New England. So Amelanchier in New England is a tetraploid. Down in the Southern Appalachians, endemic to that area, and only at high elevation in the Blue Ridge and Smoky Mountains, there's a diploid which has very similar morphology to what we in New England know as Amelanchier levis. And so we're naming that Diplo levis, as you can see in the title there. And likewise, Diplo sanguinea is an endemic to the Southern Appalachians. And a similar situation on the Northwest coast, California and Oregon, there's a lot going on there floristically, and not just in Amelanchier. There's lots of new species being discovered there all the time. It's really a hotspot of what are called paleoendemics. And in Amelanchier, pallida was known for a long time. It's an old name in the genus, but we discovered diploids that looked similar to pallida, and we're naming that diploid.
So I guess it's probably 2013 or so, Christopher Campbell and I were looking through herbarium specimens that we had on loan from Oregon. And there was a whole, probably 1 or 2 folders with 50 or 60 plants that were labeled as Amelanchier pallida, which were clearly something different. And one of the neat things about working in Amelanchier after you've done it a while is you get a pretty good idea when something is a new diploid. So you can't always guess when something is a tetraploid. So for instance, there are tetraploids that mimic diploids, and so they can be kind of hard to tell apart. But when you have something that's a solid diploid, either in the field or in a bearing specimen, you kind of know it right away. And so Chris and I looked at each other and said, here's something new in California that no one knows is there. They're calling it this completely different species. And so I flew out there the next year and collected, and sure enough, it was a new diploid, which at the time of this publication, we labeled Alnifolia var semi-integrifolia, which was the name that most closely resembled what we had in hand, even though we knew at the time that we probably published a different name. And so that is probably going to be called Amelanchier diplosiskiyuensis. How's that for a mouthful? For the Siskiyou Mountains out there, or Diplogracilis. So there's a little bit of conflict with those 2 types, which we're still ferreting out. Either way, it's still a long name. And so just as a quick aside here, these polygons that are colored on the map, where it's gray, that shows the overlap of diploid ranges. And also, as it says here, we're recognizing Alnifolia and Humulus as conspecific, as Alnifolia, so one species. So that purple and yellow polygon you can combine in your mind into the range of one species, one taxon, which is pretty wide ranging latitudinally. Sorry, longitudinally. So just to give you some global context, there's 13 diploids globally. Most of the complexity is in North America. That's because there's more species there and they're more abundant. So there's a lot more chance of overlap, a lot more chance of hybridization, etc. So I'm going to kind of gloss over the old world. So just to show you in that 2015 publication, one of the figures that was a combination of combined ETS and ITS data just shows you pretty cleanly how species in the genus segregate out. And so if you've read older papers by the Campbell Lab, you know that these corresponded to old clade names, A, B, and O. So A was for Western North America, B for Eastern North America, and O for the Old World. But as you see, A includes Alnifolia, so it's not strictly Western North America because that taxon spans from east to west. And so when you start looking at floras or papers going way back in time, these are the kind of the comments that you'll find. So it boils down to no one knows what's going on and no one agrees about what's going on, which is always fun in the field of plant systematics, right? And so part of the issue here has to deal with these differences in ploidy levels and how they interbreed with each other or not. So the graphics that I have here with these confusing circles, I know there's a lot going on. This is showing what would happen if it was a strictly sexual system, which Amelanchier is not, but just to kind of as a starting point to make it easy. So if you have a strictly sexual diploid, all in yellow there, then it pretty happily proceeds as normal. It makes haploid gametes, and they fuse and produce the next diploid generation, pretty standard. So tetraploids from a diploid can be produced in probably the rarest, sort of mathematically unlikely way. You can have 2 separate diploids that have unreduced gametes, both of them, that then fuse to produce a tetraploid. The tetraploids in some plants that are strictly sexual, let's say that you have a large diploid population that a tetraploid then is produced by the way it was described. It's a minority on the landscape numerically.
So it's produced from diploid parents. So if the diploid population is large, then that tetraploid plant or few plants, they're outliers. They're kind of the numerical oddballs. And so what genetic theory tells us is that if they're sexual and they're attempting to breed back to diploids and vice versa, then those tetraploids are at what's called minority cytotype exclusion, that they don't have enough mating partners. This is just simple math or genetics here, that they quickly become extirpated on the landscape. And so, or if there's a strong selective force and a superior genotype, they could persist. They could potentially theoretically outcompete their parental diploid or just go off on their own in a new niche. So that's pretty easy if it's all sexual. The problem with Amelanchier is it's not all sexual. So starting back in 1985, based on reports of asexuality in other genera in the apple subfamily, Chris Campbell and others researched if this was happening in Amelanchier. There's a strong suspicion that it was, and there were previous literature suggesting it, but there was never any experimental proof of that. And most malloids, the apple subfamily, the type of apomixis they have, which is asexual seed production, it's a little strange because the seed is produced asexually, technically, but pollen is needed to initiate that process. So pollen is needed to initiate growth of endosperm, which the embryo is going to use for nutrition as it develops. If that doesn't happen, typically the seed aborts. So that's called pseudogamy. So pseudogamous apomix, they require pollen still, and they're often fertile on the pollen side, even though on the egg side, they're almost all entirely sterile. Or sorry, asexual, I didn't mean to say sterile. So on any given landscape, the result of this is that you can have normal sexual diploids capable of interbreeding with each other, no problem. Next to them, or in adjacent landscapes, or literally side by side in some cases, you can have tetraploids that have high levels of asexuality. So there's about 97% of almost obligate apomixis, so highly facultative. That's on the seed side. Those same plants, though, have high levels of functional pollen, so they can still be pollen donors. And so these sort of mixes of sexual diploids and these tetraploids can produce triploids in the landscape, which are largely sterile, but not entirely. So they can also contribute back to these crosses between diploids and tetraploids. And they can all hybridize with each other. And some of those offspring and those hybridization events, they can persist for a long time in the landscape. And that, in number 5, that's sort of the biggest problem for us in Amelanchier is the persistence of these hybrid-derived entities, especially if they're small, geographically restricted, and may not have longevity in terms of a species. And then those we call microspecies. And I'll show you some examples of those. But one of the things that's most interesting to me, and we shouldn't forget this, is that, and this is true of all sciences, of course, but systematics is, I think, deeply mired in history. And if you go to the Angiosperm Phylogeny website, the very first sentence, below the header, is this line: "Systematics is a profoundly historical discipline, and we forget this at our peril." I love that quote. And it's true that there is a lot of historical precedence for how species are treated and how other experts in certain taxonomic groups influence subsequent workers. And, I'm not sure if that's good or bad. I don't think we can ever truly divorce ourselves from that. But for us, that does come back to one of our underlying goals, is that we don't want to overburden the taxonomy. We don't want to upset the apple cart. We don't want to have something so new and so foreign and at odds with previous classifications that it's unusable. As long as we can match that with our data, then we feel like we're okay. And so I tell this to my students a lot. So remember that there are certain things happening that we see, these first 3 things on this list: reproductive isolation, selection, and speciation. Those are biologically real.
I mean, our names for them, of course, are made up, but they're real processes that are happening. 4 and 5, that's not the case. So applying species concepts and classifying them, that's a totally human construct. And that human construct has been going on for thousands of years, as we know, and probably much, much longer than recorded history. Humans are natural categorizers. I mean, if you watch any child sort of grouping blocks from a young age, that's what's going on, right? Grouping things, classifying. And so depending upon your viewpoint and what you view as important in terms of how you weigh the data, that affects your classification. And so there's all sorts of practical things to think about if you yourself are the classifier. Are you a monist or a pluralist? Do you believe that there is one unifying species concept that should apply to all of life? And to many of us from, in this point in time, that almost seems crazy now, but that was a mainstream thought for much of biology. And it's still a powerful thought in biology. So if you think of one of the most extreme cases, the biological species concept, in the strictest interpretation of that, individuals can either successfully interbreed or not. If they can't, they're different species. And some people believe that species concept should be used to define all of life. Of course, that completely skips asexual organisms. Bacteria are a problem, but also many plants that have high levels of interbreeding just wouldn't fit that. So a lot of botanists tend to be taxonomic pluralists. There's a really rich literature about this, not in botanical journals usually, but in philosophical journals. And there's a couple books out by, I'm probably going to butcher his name here, but it's by Arashevsky down on the bottom. And I really recommend reading those if you're interested in the philosophy of speciation and the philosophy of how we have applied species concepts historically and will do so in the future. And then for us, there were lots of things, lots of criteria that were a little more important. Like in number 3, for microspecies, we have wrestled with this idea, do taxa, in order to be named a species, do they have to occupy a certain geographical size, a certain numerical abundance on the landscape? Do they have to be repeatable across the landscape from place to place, that sort of thing? And, and lots of scientists believe that anything above the species level is not a biologically real entity. And so there's all sorts of philosophical nuance and rich arguments in literature about this over the years, quite heated at times. So just to show you some examples of this, these hybrid-derived entities can be huge. And by that, I mean geographically huge. And so plants that look like Amelanchier utaensis are some of my favorites. I love the species. This is why I'm showing it. I know it's not in New England, but it's one of my favorite ones. I love being out west this time of year. I often fantasize about being out in southern Colorado. But if you look at the green, this is a shapefile that I pulled in and made this map of the data from Little, which is available as USGS shapefiles. Trees of North America, that volume. And so this map, based upon Little's data, shows that Amelanchier utaensis is very widespread in Western North America. But if you look at the red dots, those are, and we've been everywhere where this green polygon is. We've sampled a lot and looked a lot. And the red dots are the only spots where we have collected diploids that correspond to Amelanchier utaensis morphology. So that implies that all the green is something else. Oops, sorry about that. Which is indeed the case. So here's sort of a map of some of our sampling from that same area that Little showed, but here you can see anything that's not a red circle is not a diploid. So the red circles are the diploid utaensis, everything else is a ploidy level. That's either triploid, tetraploid, or unknown, but does not correspond to diploid morphology.
And really the center of the distribution of Utah-ensis in terms of its abundance in the landscape is really the Grand Canyon and Zion, because even though it's everywhere there, I didn't have a collection permit like I did in the Grand Canyon. That was one of the tough things about this project. I was forced to go to the Grand Canyon twice. My wife will not let me live that down. But everything else, quite wide ranging to the north especially, is tetraploid. And so this will become important because people have struggled with variability in the genus, and variability within, a valid species, even ones we're recognizing, and then variability that bleeds out of a species into other species. And so it's hard to know where the line is between species A and species B. So to show you just how one species can be variable, I want to show you these populations of Utah-ensis. So there's a taxon, Amelanchier nitens, which we're synonymizing with Utah-ensis, which is that taxon as a distinct taxon, if you were to recognize it, basically is only in the Spring Mountains of Nevada, which is just 40 minutes or so outside of Las Vegas. But it's probably, and it definitely is the lowest that I've seen. It's the most desert adapted taxon in the genus. And so this particular population is growing right with Yucca brevifolia, Joshua tree, things like that. Lots of high desert species. And locally right there, you can see it's glabrous, no hairs, kind of shiny leathery leaves. And these pictures on the upper right, upper left, and lower left are of that low desert population. But if you just go up a few hundred feet at most up the slope, you run into what's on the lower right. It's the same species, same taxon, all diploid. It starts to get a little bit hairier. So the only difference is presence of hairs going from super low elevation up the slope a little bit. And then in that same general area, New Mexico, northern New Mexico, southern Colorado, parts of Nevada, and southeastern Utah, there were a lot of named taxa historically, which turns out are all diploid and all, we're synonymizing them all with Utah-ensis. And they differ subtly, if you can see, in leaf shape, like this one's a little more crenate. This one down here at the Utah-ensis type locality is a little more orbicular. The one over here, rubescens, it's a little sharper tooth, etc. But we know, if we look at ourselves, for instance, we know that any sexual species has some inherent variability. But for some reason, botanists and biologists in general, they haven't liked that variability historically, and they've been tempted to name as species minor differences in the landscape. And that's actually what's happened a lot in Western North America with this genus. So if you can see here, just looking from these glabrous kind of leathery leaves on the top, going to something a little bit hairier here, the only real differences is pubescence. And so we have no observed differences in our morphological data sets for any other characters for this species. The other side of this, though, is sometimes people have taken this the other way and they've extended the morphological variability or their idea of the species too far. And so this is Amelanchier mormonica, which is around Mormon Lake in Arizona. And all our morphological and DNA analyses show it to be a hybrid between Olifolia and Utah-ensis. It's been historically named as its own distinct species, but most of the time it's been lumped in by other authors into Utah-ensis. So that lumping into Utah-ensis obviously greatly widens the circumscription. This leaf in my hand here is nothing like the leaves that I just showed you a minute ago. And then at certain sites, like the Grand Canyon is famous for this, because there's so much microtopography and microhabitat, you can go a couple hundred feet up or down slopes or get different orientations on the slope, and you can have roses blooming on one side, and then you turn the corner and there's a snowbank still. So because of that, you get all these Amelanchier blooming all at the same time. And so we have some nice diploid Utahensis blooming in there, down at a little bit lower elevation. I walk up the slope around the corner and I see this hybrid on the right. And then at high elevation, there's tetraploid Amelanchier pumila, completely glabrous, just basically right below the snow line. This probably was mid-May, end of May that year. So you get this syntopic flowering that's all within the range of pollinators, by the way, that's all close together, producing this just bewildering mix of intermediates. So if you look at this from a DNA perspective, a lot of these plants that look like Utahensis, quite nicely do cluster with the diploid Utahensis in our phylogenies. So everything that's in bold here on the screen is a tetraploid accession. So that was an example of something that's really widespread geographically. The other extreme, but produced by the same processes, are microspecies. So the only difference really is geographical size. So they are produced by the same hybridization processes. This typically hybridization is coupled to the jump to polyploids. So they're allotetraploid apomix for the most part, but they're narrowly distributed. And it says they're plus or minus distinct. And so, yeah, a lot of times they look a lot like a diploid species. Other times they don't, like the one that's shown on the screen here, Amelanchier rubra from down East Maine. It has a distinct morphology unto itself. It looks most like Amelanchier canadensis with some sort of levus crossing in there. But even though it's distinct, it's very narrowly distributed. And there's no evidence that these narrowly distributed but morphologically distinct microspecies have longevity. In other words, there's no clear picture of what their trajectory as a species, if they will become one, is. Now, then this is probably the biggest point of contention with this classification, is that there are some who say if it's a distinct morphological entity, they're going to name it as a species without thinking about its temporal trajectory. You could do that, but we are cautioning against that because everywhere we have looked geographically, we found microspecies. And so conservatively in this country, there are hundreds, probably thousands of them. And a lot of them appear to be ephemeral on the landscape. People have gone back to them decades later and they're gone, or something else morphologically has replaced them. So it's not clear that they have longevity like we expect species to. And this one, sorry for this thing on the bottom showing up here, Amelanchier rubra, genomically, we found it's a cross between Alnifolia canadensis and either Levus or Arborea. So it's a complex allotetraploid, which is the case with many of these microspecies. And so I don't expect you to ferret out the complexity of this slide, but it's just here to show you that whenever you run any analysis that has microspecies present, and even some of the species we're recognizing, frankly, there's overlap in the data. And so Amelanchier is well known for having narrow morphological gaps between certain taxa. And so obviously, if that's the case, whenever you run an analysis, you're going to have, graphically, you're going to have some overlap in your clusters. And so in New England, what we have are diploid species, which again, they can hybridize with each other and have, and those offspring can then hybridize with themselves back to the diploid parentals or to other diploids or to other tetraploids. But here's the ones we're recognizing in New England. And we're also recognizing these polyploid taxa. But in addition to those, there are numerous microspecies. And again, as I say, we're not going to name them, and we're counseling that others kind of take a cautionary approach. However, that doesn't mean that with further exploration and further data, some of them might warrant species status over time. We're just saying, let's take a look at each one Let's use a data-driven approach and then decide, rather than proactively filling all these journals with a bunch of published names, which someone in the future would have to sort out, which is a lot of work. Okay, so just for a local example from near my part of the woods, here's just one small part of Maine. And so in the red dot, that shows you the range that we've collected Amelanchier rubra from. It's been collected for a few decades, but a lot of times when we've gone back to sites where it has been known from, it has since disappeared. Now, a lot of species, you could argue, have metapopulation dynamics, and one population might wink in and out over time, and that's certainly true. But we think this might be another indication that these microspecies may not have longevity. So just to show you this one in the yellow box here, Ligoflora, going back to that map for a sec. And if you look at all that clustering, that's around Orono. And of course, as you know, that's what the University of Maine is. So there's the old joke about bryology that mosses don't grow anywhere near except universities. So there's a disproportionate amount of collecting where taxonomists live. That same thing is true for tracheophytes as well. So we happen to see a lot of microspecies where we lived and worked. And so that's true of everyone else in other areas. And when we traveled around, we've seen the same thing. So if you were to name all of them, the amount of endemic species in the genus would be far higher than any other genus on the landscape. Probably any other genus that I know of in North America. So one of those that's around the Orono Maine area is this tree-like taxon that's called a Ligoflora. And if you look at any flora you pick up, it's going to key out to Arborea because it's something that's a tree. That's not a shrub. And it has leaves that are hairy at anthesis. That's all well and good, except strict diploid Arborea has a lot of traits not like this taxon. So it has strongly reflex sepals versus ascending like this one has at anthesis. It has narrower leaves, less rounded leaves, and generally narrower petals. So to call a Ligoflora Arborea is to greatly widen the circumscription. And that has been the historic case for many of these taxa. So you might say, what do you do? Okay, so one of the extreme approach is because, as far as we know, all diploids can hybridize with each other, and we have actual evidence of many of them crossing. And we also know that there's a high level of fertility amongst the entire apple subfamily. So Amelanchier can produce hybrids with Aronia, produce hybrids with Sorbus, and some of those other genera can then cross with other things in the apple subfamily. So it's been seriously proposed by some authors that there should just be one species in the genus, that everything that we're calling species are just subpopulations of one taxon. And that seems pretty extreme because that's ignoring any sort of ecological differentiation, any phenological differentiation, etc. And this kind of ties back to how we view species in the first place, is that even though we all know speciation is generally a prolonged process in most organisms, we want it to be done. When we look at the plants or animals, we expect them to fit into nice categories. We don't like to think we're not seeing the finished process. So there's a lot of human psychology going into some of these classifications, I believe. So, So a first more rational option would be to name different cytotypes. And so there was a growing consensus in botany that cytotypic differentiation is one of the first steps for speciation. And you can have two plants that look morphologically similar, but if they differ by ploidy level, then they probably are on at least quasi-independent evolutionary tracks. And so they're often naming them as species. And so the biggest pro of that is that you are recognizing distinct gene pools, which are the evolutionary units. But it's really difficult to, in a real world, to use. As a lay person learning the flora, even as a professional botanist, you all don't have access, and I don't now have access to things like a flow cytometer or DNA sequencer. So it's really hard to get the data you would need unequivocally to say what the plant is. So the historical approach is, if it looks like species A, it is species A. And so that's been okay with humans for a long time because it's really easy to use. The downside of that is that there are cryptic species hiding in that now wide circumscription. So if you make your bin so big, yes, it's easy to fit things into, but you're probably overlooking some real diversity that exists, both morphologically, ecologically, genetically, etc. And so, again, I don't expect you to see the fine details of these graphics, but just to show you that this second option, which again is the common one historically, the result is wide species circumscriptions. And all of these graphics, and what I want you to look at is the different colored circles and squares represent taxa collections of each species. Let's look at the biggest one, down in the lower right, Amelanchier arborea, for instance. Those gray circles are the diploids, triangles are triploids, and the blue squares are tetraploids. So this just shows you that tetraploids are quite a bit bigger in those characters that are measured here, which are floral characters. And if you have that wide circumscription, then your species can become so wide that you then start accommodating a neighboring species into that bin. So it's not much of a stretch, and this was done historically actually, to call Amelanchier arborea and Amelanchier leavis the same species, for instance. So what we're recommending is a third approach, and this is to still have some bins, and those bins would be species complexes. And so those complexes would include the pillar diploid species and other perhaps unknown ploidy level plants that more or less conform to the morphology and generally the ecology as well of those diploids. The biggest plus to this in our minds is that the end user, the field botanist, could identify plants to the level of their confidence. If they know 100% of the time that it's a diploid species based upon the data set that we're going to publish or data they already have, then that's great. If they're less confident, then we're suggesting they put it in species complex, but not give it a species name on the collection label. And so our hope is that this will start lessening the large degree of misidentifications that exist in herbaria, and it won't force people to make what may be an erroneous choice. The big downside, though, is, but this is just a function of the biology of the plants, is that a lot of plants are not satisfactorily keyable. So to give you an example of what this might just look like graphically, so for one complex here, this is the Canadensis complex. That complex has 3 diploid species, Arborea, Canadensis, and Diplolebus, that Southern Appalachian taxon. And I didn't put in there, just for graphical simplicity, the 4X tetraploid leavis that we have here in New England and to the south as well. The take home here is that all 3 of these diploids in the complex, they can hybridize with each other. That's what all these different colored circles on the inside of the big gray circle represent. They can also share alleles outside of their complex with other complexes. So for instance, that Canadensis on the upper right could hybridize with Bartramiana. And then you can have gene flow coming into the complex from the outside here, from, like in this case, the Almafolia complex or the Bartramiana complex, and any possible combination of all of those that you can imagine, of course. And so it's quite conceivable, and in fact, it's very common, if you have a hybrid between, let's say, Canadensis and Bartramiana in the upper right,
the offspring typically are either intermediate between the two parents in morphology, or they strongly favor one of the parents. And that strongly favoring is more likely if there's back generational crossing, of course. It's very rare that we have hybrids that have transgressive character states that are more extreme than either parental. And so the way that we're approaching this is we're looking and finding characters that successfully separate the diploids. So here's a snapshot of some of our raw data. So this is Alnifolia and Canadensis here on the screen. And so we're trying all possible combinations of the data, including characters that are historically used to differentiate taxa, as well as some new ones. And when we find the ones that are the most effective, we're using those same characters to assign polyploids to that complex, based upon them having a majority of those character states. So here is our first scan. We have 7 characters that we thought at that point were the best. We boiled down to 5. Probably the hardest thing for the end user is what's now on your screen, is that this very admittedly does make a complex key for this part of the key. So to key to complex, because there's lots of taxa in a complex, of course, that requires, it just requires a lot longer key, a more complex key. When you get to the species that you may already know, things like Canadensis, Arborea, etc., those portions of the key, I guess we could say, are more traditional, or certainly less wordy than what's in front of you. But getting feedback on this, I guess, would be good as well. I'd like to hear from people. But in this key, we have our percentages of our sample, which you could argue is something you don't need in there, but we feel it may be helpful to the user to have some degree of confidence. How common is this character state in their sample? All right. So as I say here, we've been working on this a long time, and we're kind of the in group, in the sense that we think this is workable. It's worked for us, and it's been sort of our framework of thinking about the genus for a number of years now. And we've sought feedback from a lot of people who are field botanists already, and a lot of you probably in the audience. But we're definitely interested in still hearing again from you some more of what you feel about this approach. And primarily, when it comes down to a hand in your plant, I'm sorry, a plant in your hand. My second COVID shot's kicking in today. We are all botanists, and we love to identify. And we don't often feel satisfied until we have a 100% identification on a specimen. So personally, I do realize that that's a dichotomy that some of us may be dealing with. And so I'd love to hear from people, do you think that's a problem? Or is it beneficial to you to have an out, if you will, to say, okay, I'm not sure what species this is. However, it keys to this complex, probably has the following parentage, but on my collection label, I'm just going to say it's in the Canadensis complex. I'm not going to commit to saying it's species X. And that has obvious implications for things like plant conservation, for instance. So if you erroneously identify a plant in a state where it doesn't occur, then that has some repercussions. And along with that, would you be comfortable with this idea of these sort of nested levels of identification? Or not? Does it seem something like you would not want to use? Okay, so I think I've probably run over on my time a little bit, but as we said, Amelanchier is blooming right now, and so definitely enjoy it. It's a beautiful time of year. If anyone has any great sites, especially up in Nunavut, we don't have any from there. So if you're in that part of Canada, let me know. But I'm happy to hear any thoughts, any questions you have. Feel free to email me. I love to talk about plant systematics and speciation and all that stuff. Thanks a lot.
All right, thank you, Eric. That was fascinating. And we will jump in right away. We've got one from Bob here. are all the alleles preserved in the diploid species that then occur in the tetraploids of those same diploids?
I think I'm guessing the question infers the reverse, right? Like, are the alleles from the diploids present in the tetraploids?
Yeah, maybe, yeah, maybe.
Yeah. No, not always. And so here's the confusing thing. So largely, this is simplification, but largely these species concepts for the end user would be morphologically based, right? That's what the key is. It's morphology. However, the reality of some of these tetraploid taxa is that plants that look alike and would receive the same name by a classification often have different parental combinations that produce them. So if that's what the question was, so for population A, so intermediate is a great example. We're not recognizing intermediate. There's been it's been recognized in about half the floras and not recognized in the other half. And all our analyses of intermediate plants, they're different. Each population, one population is a Bartramiana times a levis, another one is a Canadensis times a levis, another one's an Arborea times a Canadensis. So they look pretty similar to the layperson, well, even to us, frankly. But they have different origins. So they're completely, in some cases, novel combinations of parental alleles. And that's why, on one hand, it is hard. What you can't ignore is that each population of these tetraploids is probably a unique allelic combination. So you can argue for the point of conservation, yeah, they're unique. We're just arguing at this point they don't warrant species status. Does that answer the question?
I think that's getting there. Yeah. No, I think it's an excellent point about the tetraploids being each one potentially being independent origin of that combination.
Yeah, and that's kind of the norm, actually.
Yeah,
that's fairly common.
Yeah. All right, so we've got a couple questions here from Russ. Let me see. So this gets over to sort of the characteristics of the species, and it's wondering if anyone's evaluating amongst the species, subspecies, hybrids, in terms of like values like fruiting abundance, fruit size, fruit flavor, sort of more applied aspects of breeding or fruit quality for consumption.
So in terms of commercially breeding them, we need to move to Canada. They're ahead of us there. So there are active breeding programs in Canada for fruit production for Amelanchier. In my experience, Amelanchier lucida is by far the best tasting one, and that is here in New England. Utah-ensis, all the populations, well, usually it's so dry, fruit sets poor, they kind of shrivel up. And they're a very large seeded fruit. They're not like our eastern species. So those, in my mind, are not worth eating. Amelanchier levis is very good. I find a lot of the horticultural varieties that are hybrids of Amelanchier levis and Amelanchier canadensis, and there are several of those in the trade. They tend to fruit, so they're tetraploids. They tend to fruit heavily, and they're good for taste and quality.
Great. We had a question here from Diana Yolis from Plymouth State, and she's asking, what are your thoughts on using species complex as a formal taxonomic rank to be published formally with authority?
Short answer is I don't. That's kind of flip, I know, but there's over 100 names in Amelanchier, and that's just one genus. And trying to reconcile nomenclature, for anyone who's ever done it, it is a huge undertaking. In many ways, it's much more work than any of the field work that you could do. And so I'm a big believer in not muddying the waters when not necessary. I guess is my best answer to that.
Yeah. We had a question from Matt here. He says, in your talk, you were mentioning microspecies and cryptic species. Are these one and the same, or are there sort of technical taxonomic differences between what you would call a cryptic species and a microspecies?
Yeah, sorry for that, glossing over that. So by cryptic species, what I meant was that, like from our point of view, like that taxon in California, that was a diploid sexual species, which contributed alleles to tetraploids in Western North America that was unknown. And so it was geographically small, and it was lumped in by collectors with another species. So it was kind of hiding in plain sight. So that's what we mean by cryptic species in that case. And or they have morphology that closely mimics another species, so closely that you may not know the two different distinct taxa until you analyze them genetically, let's say. So microspecies we're reserving to be tetraploids.
Mm-hmm. And the microspecies would be, a single site, maybe a single clone, or
yeah, and and here, and people have talked to us and kind of argued this, and I'm sympathetic to this. There is a degree of arbitrariness in all classifications, and I honestly, this is no different. It's hard to know where to cut that line, size-wise. So there is a coastal taxon that goes from about Fall River, Massachusetts to extreme southern Maine. It's in 3 states, but it's narrow. And so we're calling it a microspecies. Others might say that's big enough geographically. So yeah, it's a slippery slope, but I guess we can say just broad, generally narrowly restricted.
All right, and then Caro asks about the Amelanchier sanguinea, that it's listed in Massachusetts, but she's wondering from what you're saying whether these plants are even that species or that taxon.
Yeah, so I probably should have shown that one instead of Utahensis, but Utahensis is my favorite, so I can't ignore it. But that's probably going to be the biggest shakeup to this taxonomy, is we're not recognizing sanguinea as a taxon. The thing that's in the Northeast. It's a tetraploid, and in all cases that we've seen, it's iterated again and again by different parental crosses. In the Southern Appalachians, there is a taxon, Diplosanguinea, which is very similar morphologically to what we call sanguinea here. But it's highly restricted. It's a narrow Southern Appalachian endemic. Now, I don't know if you saw the plant that I had as my background on a lot of the slides. That was a plant that was taken from the Mattawampig River in Maine. And that's a plant that would key out in regional floras to sanguinea. However, it's quite a bit different from sanguinea that I saw last year in New York, which would also key out. So they have some similar characteristics based upon all the floras, but the stuff in New York that I collected last year, it's growing in the middle of literally a population of diploid Amelanchier alnifolia next to Amelanchier laevis. And when you cross those two, it produces Amelanchier sanguinea. And very convincing, like the morphology looks a lot like the southern diploid. I mean, I think if I didn't take the pictures myself, and if you mix them up, I might be hard pressed to tell which was which. And so that same thing is happening a lot in New England, like different parental crosses are producing morphologies that look like sanguinea, look like the historical understanding of that taxon in New England.
Yeah. So that raises a question about the sort of the biogeography of this group. I'm not sure if you mentioned it, it might have been sort of depicted in some of the maps that you showed, but is there any trend in diploids being more prevalent to the south, and then seeing a higher proportion of hybrids and tetraploids in sort of post-glacial landscapes in the north? I know that in some other groups that does happen.
Generally speaking, yes. There are some far northern diploids. So Alnifolia goes fairly far north, and Bartramia goes pretty far north. But as a general rule, we can say pretty safely that's a yes. And there's definitely more tetraploids to the north. So when you go to the Southern Appalachians, not that there aren't hybrid swarms, because there are, but I mean, New England, every time I could go to my car and drive on any road around here and see something like, don't look at that, put the blinders on, it's something new, sort of thing. That doesn't happen as much in the South. So we have a high preponderance of tetraploids and tetraploid crossing with each other
here in New England.
Mhm.
And it gets worse when you go like northeastern Maine is probably, and down East Maine are some of the worst areas in New England for complexity. But Amelanchier is a disturbance mediated genus. And so hybridization happens the most when we, or glaciers, whatever,
Mhm.
natural or otherwise, there's some big disturbance. That's when things get crazy. And so repeated harvesting, cutting, agriculture, and now moving plants around on purpose to horticulture. Like I was saying in a talk a few weeks ago, in north central Maine, in a parking lot in a Walmart, planted in the median strip, I've seen Amelanchier utahensis. It was surviving. It was horribly diseased, but it was flowering, and it might be spreading its alleles into So whenever you have anything like that, you really uptick the rate of polyploidization.
Mhm. So Bob wrote in again to sort of reframe that question he had asked early on about genes and diploids and tetraploids. So he's saying if, for example, if all the tetraploids disappeared from the landscape, would all the current alleles still exist in the remaining diploids?
Yeah. Yes. Yeah.
So there's no evidence of species that have gone extinct and only persist, in their hybrid or polyploid offspring.
Not from our genetic sampling. No, what's interesting about that is before we discovered that new diploid in the Northwest, the way that our trees were compiling is we theorized that there was a missing diploid.
Mm-hmm. Yeah.
Some of the patterns, like, something else is going on here. We're missing something. And my personal sort of pet belief was it's in the mountains of Mexico, which there still could be something there.
Mm-hmm.
But it was kind of nice because we had this inkling that something was missing. And then we saw those ovarian specimens, went out there and collected it, and it was the missing diploid.
Yeah.
So as far as we know, I don't want to say 100%, of course, if you're never there, but we feel pretty confident that we have the picture.
You found all the diploids and
Yeah.
Yeah. All right. Let me see. I think that is getting through the questions that have been submitted so far. If anyone has any follow-up thoughts or comments, you can add those to the chat. Art had posted a question that was sort of similar to the one I had asked about diploids in the South. It may be a slightly different take on this, but he's sort of wondering about the persistence of some of these diploids in small regions, refugia, and then the widespread complexes or tetraploids. And is there, I mean, I could maybe sort of expand on that and just like, what is it about the tetraploids maybe that it contributes to there being more widespread as compared to some of these diploids that have very small ranges? Is there anything about the sort of being a polyploid that would contribute to greater expansion of ranges? Or is it just the repeated formation of them in many different locations so that they appear wide-ranging, but they're actually multiple origins?
I think it's both. So I think multiple origins does play a big role in it, but we know that polyploids, of course, they're freed from needing a pollinator.
Yeah.
Right. So that alone allows them to spread more quickly in the landscape. Disturbance is a big player. So that little narrowly distributed diploid in Northern California, for instance, it's amazing. You can see it. It grows on serpentine, which I didn't mention. So it's kind of a unique substrate for the genus. But literally, you have intact habitat. And I guess you can't see what I'm pointing here, but room one, intact habitat. And then next to it, they've done some bulldozing and brought in some non-native soil, made the road. You have diploids in the native habitat on the road edge where they have messed it up, literally feet away are triploids. So as soon as we start disturbing the landscape, that's when polyploids take advantage of that niche space. So they seem to be more disturbance adapted. The diploids seem to be better competitors on intact substrate, especially that's adaphically different. So in the South, it's in the West, it's things like dolomite and serpentine. And down South, there's something called amphibolite,
that
Mhm.
Diplosanguinea grows on. So they seem to have the niche space kind of carved out where they perform preferentially better on than the tetraploids, and vice versa. Tetraploids
Mhm.
do great in disturbance.
Cool. And in some of the work you were showing that was looking at sort of the global distribution of this genus, has any of that work indicated potential areas of origin and directions of migration over millions of years? Does this genus appear to have originated in North America or Asia and then migrated in one direction or
Yes,
the other?
yeah. So most of the data, there are some different viewpoints on this, but the majority opinion and most of the data supports the fact that it originated in this, not North America, but probably Central America and spread north. So it's the genera two most closely related to it are Paraphyllum and Melicomeles. So Paraphyllum is Western North American, Melicomeles is Central and South American. And so we think it evolved there, spread north into North America, and then colonized the rest of North America and Eurasia.
Okay, cool.
There's some fossils from Western North America that are about 60 million years old, which look very, very similar to Utahensis that's on the landscape today.
Oh, yeah.
We've been around a while.
Yeah. We've got one question from Stephen here about whether there's differences between the Eastern ulnifolia versus Western ulnifolia.
Not really, and so that's why we synonymized them into one species. So as you know, it was treated as ulnifolia and Humulus for many years. It largely was based upon geography and confounding it with tetraploids. When you look at just the diploids across that range in our data set and all the analyses that we've run, both morphologically and genetically, they're the same thing. Yeah. So no discernible differences, other than normal things you would expect in any sexual population.
Yeah.
But statistically, no differences.
You had some interesting comments when you were talking about species and microspecies about sort of the persistence of species and their longevity and I'm wondering if any of the work that you had done or the Campbell lab had done is detecting differences in the relative ages of the diploids versus some of the tetraploids. I'm not sure if that would be something that would come out of those analyses.
Yeah, that's always intrigued me, but no, just the data that we have, the type of genomic sequencing that we've done just does not have the resolving power to get at that question. I wish it did. That's really something that I think should be looked forward into the future. Now with whole genome sequencing, it might be easier and easier.
Mm-hmm.
That's a fascinating avenue of research, I think.
Cool. So Paul is asking, what factors contributed to recognizing Nantuckensis?
Oh, sorry, we're out of time. So that's a good question. So there are some species like that are polyploid. And so here's the approach that we took. If they're fairly widespread, which that species is, and it has a distinct, repeatable morphology, then we recognized it. But as I say, it's a slippery slope, and that could be construed as sort of the point of arbitrariness there. And so tied into that a little bit is sort of our willingness or unwillingness, I should say, to upset the apple cart too much. Most authors have recognized that as a distinct taxon, certainly as a distinct morphology that repeats on the landscape.
All right. Thank you for presenting tonight. This is really, taxonomically fascinating. It's a group that a lot of people love and appreciate in the landscape, and just getting this detailed perspective on the group is really fascinating. So thank you
Thanks for having me.
Presenting tonight.